Assalamu alaikum wa rahmatullah. Peace be upon you all, and welcome to another yet very exciting Finding Truth podcast. And today I'm really honored and super glad to have with me the renowned Dr. Michael Behe. And first of all, how are you doing today, sir?
Oh, I'm doing great. Looking forward to the conversation.
Thank you. And of course, if somebody has been, on a trip to Mars or something, and you don't know who is Dr. Behe, I'm going to give you a little introduction, which will not do him any fairness, it will not do him any fairness, but just a short one. So Dr. Behe can actually be seen as the father of the intelligent design movement and the one who coined the term irreducible complexity. Which is what we're going to actually discuss today, among other topics. Dr. Behe is a professor of biochemistry at Lehigh University in Pennsylvania and a senior fellow of the Discovery Institute Center for Science and Culture. He is author of the landmark books, Darwin's Black Box, The Edge of Evolution, and Darwin Devolves. Okay, and the one that actually addressed the irreducible complexity is the first one. Isn't that right, Dr. Behe?
Yes, that's right. Way back in 1996.
Yeah, so since 1996, it's like more than 25 years now. So it's like 26 years. And it's been quite a trip, Mike, right?
Yeah, it sure has. I didn't quite know what I was getting into, but I found out soon enough.
I am sure no regrets. I'm sure no regrets. It's for sure to me. I am sure thousands, if not tens of thousands, if not hundreds of thousands, have had illumination from your writings and your ideas, and many people who would have otherwise lost faith or got deceived by the mainstream propaganda have been saved from it. So first of all, thank you for all what you have done, and I hope today that we shed some light on your ideas. And because one thing that I have noticed is that I think the ideas were so good to the extent your opponents couldn't actually intellectually counter them. So all what I have seen is like saying, oh, irreducible complexity has been debunked. This is no evidence, defaming things, misrepresenting things. So hopefully, what we will try to do today is essentially to shed some light to make sure people really have some quality time understanding some of those concepts and understanding why, at least in my opinion, I'm sure also in yours, none of them has been actually debunked. And to me, every single day that passes, I see reconfirmation after reconfirmation of your splendid ideas. And the reason I really love them is that they are actually engineering ideas. I'm not sure if you would actually agree to that. But they are actually,
Engineers generally understand irreducible complexity a lot better than biologists do.
Well, so that's why they're very, very close to my heart. And I, before myself personally getting exposed to them, I used to use the term integrated systems to express the same thing. Sure. And then I was really happy to find your material, and that was a long time ago. And wow, the term reducible complexity actually adds more insight. It's not just an integrated system that if you remove one part, but actually wouldn't at all work. So let's start from the beginning. Let's start because, as I've said, you are the father of intelligent design, let's say. But let's first take the first word. When we say intelligent design, the first question is, what is intelligence?
Well, it's hard to define, but I think a good way of looking at it is the ability to put things together or to arrange things for a purpose, because only intelligences can have purposes, and to express their purposes, they might choose things. Intelligence, the word intelligence means to choose between. So they can choose different things to advance their purposes.
Okay, and so when we say there is intelligence behind life and the progress of life, then it means there is a mind that's taking decisions. It's not just like flowing on its own. Would that be a fair representation to the?
Yes, that's right. It is arranging things for its own purposes, and so it has purposes. That means it has a mind, and we can, yes, we see this all the time.
So if we take it one step further, how would we define ID or intelligent design as a framework? What is intelligent? When we say that this system is intelligently designed, what would that mean? And then it means, how would we recognize if something is intelligently designed?
Well, intelligent design is best defined as the purposeful arrangement of parts. I just said that intelligence is the ability to have a purpose and to take steps to fulfill it by arranging things. Intelligent design is essentially the fulfillment of that. A mind has arranged stuff, and we who also have minds recognize the arrangement or the purpose behind the arrangement, and so we perceive purposeful intelligent design by seeing a purposeful arrangement of parts. The one example that I use a lot is a mousetrap. And anybody can tell that a mousetrap is designed because you see a bunch of different parts and they all work together and they can do something. So we see this in life as well. So that's the idea of intelligent design.
I just want to ask an extra question here. If we find something that is like carved or shaped, let's say we make a hammer of one piece, like we make a mold and we put an alloy there and it's like in the shape of a hammer. So it's like one part, but we can still recognize that it is intelligently designed for a function, be it, driving nails in a piece of wood or something. So is it necessary that a design would be multi-part, or even your example of like Mount Rushmore, where it's still like one piece of big rock, is still intelligent design? It's like
that's right. Yeah. That's a very, very good question, and you have to be careful of how you define a part. A arrangement of parts, because if you're shaping plastic so that it can be made into a hammer, you have to make the mold, which goes up and then it turns a little bit and then goes up and goes into the shape of a hammer. So each decision there to have the shape just right, you can say is a purposeful arrangement. So and same with Mount Rushmore. It's one piece of stone, but this piece is, angled at that. So you should consider them separate parts. The nose and the eye and even the shapes of the nose. So it's not just it's one continuous physical piece. You can always say that anything would be one piece. Even say a mousetrap, you might say tie a string around all the pieces so that somebody would call them one piece. But it's all of the significant aspects of those as well.
All right. So even if somebody tries to go around that, at the end of the day, when we look at something that is purposefully altered, we immediately recognize its design, rather whether the alteration is putting parts together or actually taking one piece and carving it or shaping it or molding it. At the end, maybe it is the purposeful arrangement even of the atoms of the thing. Then the atoms or the molecules can be the parts at the end.
Sure, yeah? Absolutely. And that's certainly a topic that comes up in life, in biology, and when you go to the foundation of life, molecular and cellular foundation.
Yeah. So, for example, when you look at a protein, it's at the end a set of like peptides, which each one can be seen as a part, right? Or amino acids, each one can be seen as a part, and then that they are in this arrangement or in not any other arrangement, and that this specific arrangement does a specific function, will also fulfill the purpose, right?
Exactly. Yeah. The proteins, as I'm sure most viewers know, are connections between amino acids, which are the building blocks. And they're a lot like letters, the amino acids in a protein, in that you can arrange letters to make different words. And you can use the different arrangement of words to make different stories and so on. And yet you only have a few, 26 letters in the English alphabet. And it doesn't matter if they're hooked to each other. It's the fact that they are different from each other, and the identity of the individual attached pieces allows the protein to have a particular function.
So, Dr. Behe, some attack intelligent design and even go the length of calling it pseudoscience under the pretense that it is unfalsifiable, and scientific points of view or scientific theories or scientific propositions need a test for falsification. So it put forward a proposition and say it is true because so and so, and if so and so happens, it's false. So you give people an opportunity to prove you wrong, just like you are proving yourself right. So is it true that intelligent design is unfalsifiable? And if there is a falsification, can you mention how can somebody go into whether a lab experiment or even a thought experiment to prove it wrong?
Well, yeah, I think that the interesting thing is that intelligent design is easily falsifiable, but Darwin's theory is more difficult, and we can talk about that. But with intelligent design, my claim is that systems or of a certain complexity and irreducible complexity can't be put together by unintelligent processes, including Darwin's idea of random changes and natural selection. So if somebody wanted to show that was wrong, all they would have to do is go into a laboratory and grow bacteria, say, for thousands and thousands of generations and very, very large numbers of bacteria, and show that some system that has a number of different parts and does something new had arisen by mutation over that time period. It turns out that has not occurred. As a matter of fact, a man named Richard Lenski, who's a very distinguished scientist at Michigan State University, has done an experiment like that over decades, and over 50,000 generations of bacteria have been grown and allowed to mutate and change. And over the course of his experiment in 30 years, there have been trillions of bacteria. And at the end, no new irreducibly complex systems have been seen. And as a matter of fact, the most frequently altered genes were ones that were already there, and they were mostly altered to degrade them or to break them. So if Lenski's experiment had shown some brand new system, then my ideas would be blown out of the water. So yeah, intelligent design is pretty easily falsifiable.
So the falsification here would be, correct me if I'm wrong. So let's say that intelligent design is the antithesis philosophically, where the dividing line would be purpose. So to the right of the line, there is purpose and hence intent and hence guidance. Right. And to the left of the line, there is no purpose and hence no intent and hence randomness and chance. This is all what remains. Right. So to falsify the right side of the formula, you will have to show that really chance plus randomness can produce design and prove it in the lab. Design would be in what you have described now, the appearance of novelty of complex functions that did not were not there prior to the application of the unguided conditions.
Yes, that's exactly right. After all, that's what Darwinists say happened over the course of life, that all of this fantastic and intricate machinery that is found in the cell developed by random changes and natural selection. So, in order to falsify intelligent design, essentially they just have to put their money where their mouth is and show an example of such things as we watch. And it was thought that it was thought in the past that it would take too long to do so, but with Lenski's experiments and other folks working on microorganisms that can reproduce rapidly, there are very good experimental approaches, and none of them show anything like an ability to put things together for new purposes, new functions.
Actually, I would like to dwell a little over the Lenski experiment, because for many reasons. Because number one, because you mentioned it, but number two, it's because I have gone into many debates with Darwinists, and I noticed that they use Lenski experiment actually completely to the opposite of what it proves. Mm-hmm. So number one, they call it the long-term evolution experiment, where no evolution has actually occurred. And number two, they would say, okay, look, so those bacteria only knew how to metabolize glucose, but after so many tens of thousands of generations, voila, suddenly they can actually metabolize citrate. And they say, here you go, here you have it. It's a novel function, and those bacteria, E. coli in this case, they evolved this new function. So if you can shed some light on what actually happened, and so to demystify this and to show why this is not true. Yeah.
Okay. Yeah. Well, you have to, as I've dealt with this topic of Richard Lenski's work and the citrate mutation in particular in my book, Darwin Devolves, which was published a couple of years ago. And in general, the thing is that these bacteria called Escherichia coli, E. coli, were growing in his lab. And one morning he came in and saw that there were more bacteria in one flask than in any of the other ones. And so that indicated that it had somehow acquired the ability to grow better in larger numbers than the other flasks of bacteria. And so it took them a while to track it down. And it turns out that the bacteria were eating a chemical called citrate, which for technical reasons they had in the nutrient broth in which they were growing the bacteria. The non-mutant ones only ate a sugar, glucose, that Lenski and his coworkers supplied in that broth. So they were excited because it could do something that the other bacteria couldn't do. And it got a lot of attention because of that. But it turns out, it's interesting and all, but it actually just activated metabolic pathways that the bacteria already had. It turns out that even the non-mutant bacteria can metabolize citrate, but only if there isn't oxygen around. Turns out that oxygen helps to regulate the metabolic pathway, and it turns off this other pathway when it's on. And in Lenski's lab, the bacteria were always grown with oxygen present. Well, it turns out that one of the bacteria kind of rearranged a couple of genetic elements that were already existing there. And turns out that a control element from another gene, which was turned on all the time, not just in the presence of oxygen or in the absence of oxygen, kind of got flipped over and landed in front of a gene called a citrate transporter, which is turned off in the presence of oxygen, but in the absence, the gene codes for a protein that kind of acts like a door to allow citrate to come into the cell. Well, with this other genetic control element next to it, it was turned on all the time. So that mutant could pull citrate in the presence of oxygen. All of the other proteins, all the other enzymes for metabolizing citrate were already present in the cell, including the transporter. It was just kept turned off. Well, that's interesting. That's nice. And, you can, Richard Lenski's lab made a big deal of it. And I'm not surprised because, hey, you have to get grants and funding and stuff to continue your work. But I kind of viewed it as a kind of a sideways evolution, not really much of a much. It did not gain what I had in the past called a functional coded element. That is a new controlling element or a new gene or part of a gene. It just had rearranged pre-existing ones. But as I wrote in the book and I discussed on my blog, it turns out that prior to this mutation only happened a while after his experiment started. But before this, about 10 other genes had mutated, which allowed the bacteria to grow faster. Not the citrate one, this was before the citrate appeared, but just to let it grow faster than than the parent bacteria. And it turns out that all 10 of those genes had, the mutations had broken those genes. And that allowed the bacteria to grow faster. And even with this other mutation that allowed citrate to be imported into the E. coli cell, it still had all of those broken genes that it carried along with it. So it still degraded considerably from what it started out as. Furthermore, Lenski published a paper a year or so ago following the citrate mutant even further, allowing it to grow in his laboratory again, and check to see what other mutations might come along to help it take advantage of this ability to metabolize citrate. And genes were being destroyed right and left. Wow. And they would help it metabolize the citrate. Yeah, they would, but at the expense of pre-existing genetic information. So essentially the bacteria was burning genetic information for short
surviving. Yeah.
In my book, I kind of liken it to a youth who inherits a lot of money from somebody else and goes out and spends it here and there, and he's going to run out of his money pretty soon. But while it's there, he's having a great time. And
yeah.
Lenski also showed that the citrate mutants, as they proceeded, the rate of the death of the cells kept increasing and increasing. The ancestor cells had a death rate, I think it was about 5%. That is, 5% of cells died every generation or so. At the end, it was about 50%.
Wow.
Half of the cells were dying each generation. So again, as I wrote in the book, this is devolution, and it's statistical. Devolution can occur a lot faster, and it's easier. So you lose genes, and maybe once in a while you get a hiccup where you have this rearrangement like in the citrate, but it doesn't fix the previously broken genes, and the overwhelming tendency to continue to break genes continues.
Great. So I have actually 2 points to discuss further with you here. So from what you said, I think we can categorize, correct me if I'm wrong, what happened in the Lenski experiment into 2 categories. one which is breaking of genes, and another one which is a rearrangement of the existing genetic material to the extent that the glucose transport would now also work for citrate transport. So now those trucks will go to the cell membrane and carry citrate molecules from outside of the cell and carry it in, rather than just pick the glucose and carry it in. Right. And I think Planski chose to use the word acceptation for that. Actually, I had a long format thing on my channel about that, and it strikes me that if you look at the cell and you find all the machinery for metabolizing citrate already there, and only the door for citrate coming in is closed, and then when there is no glucose and there is citrate instead, then the door is open. Is it at all fair to say that this is a random event, or it is rather more sensible to say that this is a kind of adaptive response that is following a certain sensing of the environment and taking an action, rather than a random thing? Because otherwise, if all of those machinery for metabolizing citrate were in the genetics of the cell and were not used, shouldn't, according to the very premises or the very resolutions of the Darwinian thinking, shouldn't all those genes should have been lost by natural selection? Yes. Or not? What would you? And then I have seen people talking about bacteria being intelligent. So you have this divide. Some would say that this is completely random, or they would say it's an acceptation, but the acceptation itself they would categorize it as random. And then some would say there is intelligence in bacteria, but they would rather not explain it. So what's your comment about that?
Well, you can look at it from a couple of different angles, I think. One is that with the bacteria you have, the E. coli you have, does this mutation where it messes up the control for the citrate transporter, or flips it over so that it can activate that, does that occur within the probability distribution that you would expect? So is it some exceedingly unusual event that you wouldn't expect in the trillions of bacteria that are growing, or is it within the bounds? Maybe it happens once in 100 million events or something. And I think it's probably, with the starting bacteria, it's probably within just a probabilistic distribution to have that mutation. But then you can ask yourself, well, in the starting bacteria, how likely is it that we have something that could just degrade and nonetheless allow the bacteria to adapt to this different situation? And that's a lot harder to get a real statistical or probabilistic handle on. One could argue, I think, that, if the cell were designed to endure various environmental insults and go through problems, that it might be the best way to allow other pathways to take over critical functions when the circumstance demanded it. But again, I think that's a little less certain. You can't. It's not a definitive answer, and you might be suspicious that that's the case. But I think if you look at just at the machinery that's present there, without speculating about what might else happen, you've got more than sufficient material to strongly conclude intelligent design.
Great. So actually, this was the second part of the question, because it goes. What's your comment on people talking about intelligent bacteria? Why, in your opinion, they choose to talk about intelligent bacteria rather than an intelligent designer? Because obviously, when you have all the machinery, and then actually the way I remember it is that this citrate transport thing was a transposon, and it actually jumped and moved exactly to the right place in front of the promoter that used to to get glucose in, etc. So this, to me as an engineer, especially that it even happened into two different flasks or two different groups. So it seems, because random things don't typically happen more than once, very rare random things. So it drives me as an engineer, as somebody who would rather see that this is a reaction that kept this thing surviving, that it's intelligent design rather than intelligent bacteria or just chance.
Yeah.
Why would a reasonable person take the disposition that this is like intelligent bacteria? Don't know what that means, or that freaky chances happen over and over again. Wow.
Well, I think that it goes back to philosophical reasons. First of all, people are confused about what intelligence is and what consciousness is, and these days, because with the turn to materialism, following hard on the heels of Darwin's theory, people have tried to get rid of intelligence and mind and so on. So now when they come back and see the enormous detail and elegance of the molecular level of life, they're trying to sneak intelligence back into the discussion, but at a tame level where they don't have to actually think about some supreme intelligence or higher intelligence. So they'll attribute it, maybe the bacteria is intelligent, or maybe maybe the universe is intelligent, or something that strikes most people as strange. So I think
I'm hoping we don't find bacteria temples soon.
Yeah.
Worshipping bacteria, because
that'd be great.
They're intelligent. We can't live without them. Wow.
Yeah. So it's really strange the way some people think about this.
Yeah. So I guess philosophically there is always a move. What you're suggesting is that there's always a move away from guidance, intent, and purpose. So let's, before addressing Darwinism directly, when you talk about unguided evolution, what does that mean? When people talk about evolution, I think this is also a very big area of confusion to the masses, to the layperson, because you can tell them, guys, evolution is a theory and it's a false theory. They say, what are you talking about? Evolution is a fact. And I think there is great intentionality in the mainstream to mix between the fact that there is change, that there was dinosaurs and there is no dinosaurs now, and there are humans and there was no humans, which is all obviously a fact, versus how this happens. And the philosophical position, as you were saying, is that it's happening completely unguided, just with chance and randomness. So is that the case? Is that what unguided evolution is? And how many flavors does it come in? Just Darwinian mechanisms, or other things are there also?
Well, unguided evolution is pretty much what it says. The people who use that phrase generally mean that there has been no intelligence pushing things this way or that way or the other way, pointedly including God. Nothing. It developed unconsciously or unintelligently on its own. This is just no more the result of intelligence than, say, a thunderstorm is, or a tornado, or something. What was your last comment?
What flavors does it come in? So when we're talking about Darwinism or neo-Darwinism, those are two flavors. If you can explain what they are and what other flavors are there, and then if they are falsifiable altogether or not. Yeah.
Okay. Yes. Darwin proposed that it was the first to propose that life could develop non-teleologically, with no purpose, no outside influence, nothing like that. Other people had talked about evolution in the sense of descent, but it was usually a teleological, guided, inbuilt kind of process. And Darwin put up the idea of random changes They didn't know about mutations because they didn't know about DNA and molecules and stuff back then. So random changes in natural selection. And of course, the basic idea is that maybe a change comes along and helps an organism survive and leave more offspring. And then in the next generation, if the offspring inherit the mutation, then they would still have an advantage, and their grandchildren and so on. And that was Darwinism. Now, it turns out that Darwin and his contemporaries didn't know anything about genetics or the molecular basis of inheritance. And the cell was pretty much a mystery to them, a black box, I think I said. And so they just talked about changes. And they emphasized natural selection because the change part, the random part, was completely opaque to them. But then with the progress of science in the 20th century, DNA was discovered and genes were discovered. And then it was discovered that genes were made of DNA. And slowly, it turns out that the basis of heredity was kind of pinned down to elements of DNA. And so neo-Darwinism pretty much incorporates Darwin's idea of random changes, but as random mutations and then natural selection, and builds in what has been learned about heredity and genetics and so on. Now, it may surprise a lot of listeners to find out that a lot of biologists are very skeptical of Darwin's theory, just of neo-Darwinism, random changes in selection. And a lot of biologists, maybe a third of biologists, propose something different, which they call the extended evolutionary synthesis. And they say, well, maybe other things come along, such as something called epigenetics, where DNA can be modified, or interactions with the environment, where the creature modifies the environment and then is selected to live in a modified environment. For my money, none of those is even as persuasive as Darwin's mechanism. All of them, including natural genetic engineering, which I think we'll talk about later, and epigenetics and so on, still depend on doing some change, getting some change, and helping an organism, and in that way spreading throughout the population, and then coming along and building on that and again and again. It's interesting that despite being around for 20, 30 years, the extended evolutionary synthesis has not made any progress in explaining the molecular machinery of the cell or other modern molecular biological discoveries. They tend to emphasize, features of organisms like colors of fish or how termites build mounds and so on. And that's interesting. Sure, certainly that's part of life too, but it doesn't explain at all the molecular machinery that has been found at the basis of life. So I appreciate their skepticism of Darwin's theory, but until somebody's willing to recognize intelligence in life, then they're not going to come up with a satisfactory explanation of these things.
Great. So I'll just ask the counter question now to what we asked about intelligent design. Is the Darwinian / neo-Darwinian mechanism or ideology, or the unguided evolution, regardless what flavor it comes in, is it falsifiable itself? So does it qualify to be a theory? Because if it's not falsifiable, then it doesn't qualify to be a theory.
That's an excellent point, and I've written that I don't think it is falsifiable, and or it's extremely difficult to falsify. Because I wrote about the bacterial flagellum, say, in Darwin's Black Box. And the flagellum is this little outboard motor that can rotate, that bacteria can use to swim through liquid. And just like an outboard motor in our everyday world, it's got a number of different parts that have to interact with each other to produce the motion. Now, how would you, if somebody claimed that that was made by a Darwinian process somehow, how would you falsify that claim? You know, if you said, okay, let's take this bacterium and using some molecular biological laboratory tricks, we'll eliminate its ability to make a flagellum. We'll knock out all the genes that are involved in making a flagellum, and we'll grow it a long time under a selective pressure where the ones that can move a little bit will have an advantage.
Yeah.
And we'll see if it's got a flagellum back or any complex motion system. And at the end, suppose that, well, nothing was found. What would a Darwinian say? Would he say, okay, well, you're right. You know, I guess I was wrong all the time. No, he'd say, well, you didn't let it go long enough, or you started with the wrong bacterium, or you knocked out the wrong genes, or the growth conditions weren't correct, or any of a number of different possibilities. So instead of saying that Darwin mechanism didn't work, he'd say a number of other conditions just weren't correct. So again, ID is if the flagellum, the new flagellum did arise, then intelligent design would be falsified. But if it didn't, then Darwinism would not be falsified. So yeah, I think a lot of people, you know, accuse intelligent design of being unfalsifiable, but I think they get it exactly backwards. And they think, well, you know, UID folks are saying that God, you know, directly zapped this flagellum into existence, and how can we put God in a test tube? Well, you can't, but you can just show that unguided processes have this amazing ability that you already told us they do have, and just show us what it can do.
So, Mike, I have this question. It really bugs me, and I don't know how neo-Darwinism made it as a theory in the first place. I really like to tease evolutionary biologists when I go into debates with them. I just tell them, I'm sorry, this is not a theory. Your theory does not qualify to be a theory. It is, to the best, a false hypothesis. And because it doesn't have all the cornerstones of a theory. And set aside all the evidence against it, but how did they push this thing to call it a theory of evolution with a capital T? It's really mind-blowing to me. So yeah, how did it happen in your opinion?
Well, the short answer is I don't know, but a longer answer is that when Darwin first proposed it, remember, nobody knew how genetics worked. So they could use their imaginations to think, well, maybe if a creature, you know, had a snout and it got longer and longer, you know, maybe it would turn into an elephant. Without they didn't have the ability to look and see what kind of steps would be necessary to do that. And also, it turns out, from what I read, I'm no historian, but Darwin's mechanism, when he proposed it, when The Origin of Species first came out, was not well received. That many scientists of that time loved the idea of common descent and loved the notion that science could investigate how life developed, how life arose and changed. But they thought that Darwin's mechanism was completely inadequate to explain it. But then for, I guess, pride of professional pride, they claimed the right to be able to tell everybody how life developed. But even Darwin's strongest defender, a guy named Thomas Huxley. He was called Darwin's bulldog, and he would debate in favor of evolution, but he was very skeptical of natural selection as an agent. But so actually at the turn of the 20th century, Darwin's theory itself was in eclipse. That is, it was receding from view. Not many people were thinking about it. And it wasn't until Gregor Mendel's work in genetics where that was rediscovered, that it was seen that traits could be inherited discretely from one parent or the other parent. And I think he used peas in a garden, in a monastery garden, and showed that the colors of the peas matched up the genetic pattern. And the important thing, in 1930, I think the book that set it all off was written by a mathematician named Ronald Fisher, who wrote the the genetical theory of natural selection. Mm-hmm. And he used algebra to show how these genes would rearrange. And suppose there was a different modification of gene A, well, then you could have 2 copies of A matched up with B. But what if there were 2 copies of B? Then you'd have 4 combinations of the 2. And if you had 100 genes where there were 2 different variations, alleles, then you would have 2 to the 100th power possible combinations. And that was such a large number, they said, wow, evolution can do anything. But they did all that in the absence of knowledge of how the cell ran. They didn't know DNA was the genetic material back then. They might have suspected, but didn't know. They didn't know what proteins did. And so they had very little appreciation for the complexity of life. And as I argue in Darwin Devolves, the 2 variations of a particular gene that Ronald Fisher was talking about, say gene A and A prime, might be the gene and a degraded variant of it. So you have something that works and then something that has a mutation, which makes it either very poor or broken altogether. Mm-hmm. Same for gene B and same for gene C. And if that were the case, you could still have 2 to the 100th possible combinations of those, but it doesn't say anything about where those genes came from. And it doesn't say how any of them could rearrange to make any of the coherent molecular machinery that has been discovered in life, such as, say, the flagellum or the machinery that translates the genetic code in DNA into protein. And I think they were misled by their number one, their ignorance, and number 2, their over eagerness to explain things right away. And so here we are.
I was actually waiting for this word ignorance, because listening to what you were saying, it really strikes me that if somebody in my field, like in electronics engineering, for example, comes and says, okay, we will have 100 capacitors, 100 coils, 100 resistances, and as many wires as we would like, guess how many combinations of small circuits we can have? And then he would propose that if those things can, we can put them on a computer model and then let them hit each other, we will eventually, at the end of the day, get the latest or the super duper new, iPhone 2050 or something. And this is completely ridiculous. It's as if you're saying, since all books are eventually letters, let's keep striking letters with each other and putting them in the mixer, and we will eventually get out with the Shakespeare and the Odyssey and whatever. And it's completely ridiculous. And I have actually seen evolutionary biologists talking to me about products of what you have talked about, like coalescence theory, for example. And then when I personally looked into the literature, because it's essentially a mathematical model, it's very clear from the literature that coalescence actually only deals with mutations to existing genes. It doesn't address novelty altogether. So what do you call, is it just ignorance, or it is something else that people will use theories that can be applied to neutral or degradatory mutations to existing genes and then extrapolate them to claim novelty, while this is not the case? Is it just, I don't think this is just ignorance. It is
well,
because
it gets complicated again. Certainly a large part is ignorance. Another large part is ambition. People want to be able to explain this. And then later on, another large part is sociology, because the students who will become the next generation of professors are taught by the people who believe this, and everybody in their community believes it. So it seems like everybody knows this. It doesn't even need an intellectual defense. But I think you can tell there's this undercurrent of dissatisfaction because of the extended evolutionary synthesis. People still believe in evolution, but they don't know how in the world something like this could have occurred. So it's a complex problem, but it's not hard to see. The problem isn't hard to see if you're not, kind of identifying with that academic group.
Yeah, since now you've mentioned the word complex, let's now go to complexity and irreducible complexity. I'll just need to let people watching us know that we are going to have a Q&A session at the end. So just put your questions and tag me. So complexity, irreducible complexity. What is irreducible complexity?
Well, irreducible complexity is just a system or machine or something where there are a number of different components, a number of different parts, and the parts act upon each other to produce some function or effect that none of the parts itself can produce. And if you take one of them away, then the system is broken. The function of the system can't be carried out. And in Darwin's Black Box, I used the mousetrap as an example. Even a mechanical mousetrap needs a number of different parts. I don't have an example here with me, but it's got a wooden platform and a spring, and it got a hammer which squashes the mouse, and there's other metal parts too. And they've got to be arranged just right, or it doesn't work. If you take away the spring, if you take away the hammer, it's not like it works a little bit or half as well as it used to. It's just broken.
It will just not catch any mice. Yeah,
exactly. Yeah.
It will cease to be a mousetrap.
Yeah, the mice will dance around it and not care.
Probably enjoy the cheese.
Yeah. Well, it turns out that most machinery, almost all machinery, is the same way. A machine is something that has a number of parts in order to perform a function, oftentimes to, redirect force or other things. And if a crummy mousetrap is irreducibly complex, then certainly more sophisticated machines will be too. And in Darwin's Black Box, I talked about the flagellum and other systems where if you do take away one of these parts, they are broken. So it's irreducible complexity is something that's complex, means it's got a number of different things going on there. And it's irreducible in the sense that if you take one away, then you don't have the machine anymore. You don't have the function.
I will just want to just make something clear here to some people, because I have seen some of your opponents, come up to debates with you with some absurd propositions, and you are really so polite and so calm. And I remember one of them used a mousetrap as, I don't know, a clip for his tie or something, or
Tricolor,
something like
right?
that ridiculous. Someone else said I can use it as a paperweight or something. So
Yeah.
is it that they do not understand what irreducible complexity is about, or they're just because okay, I will just not even ask you, because if I'm using a mousetrap as a paperweight, obviously it's not a mousetrap anymore.
You know, yeah, yeah. Well, I think what you said before is that such folks just don't have answers to these problems. It's because they're looking at it the wrong way, and so they come up with these, easily debunked ideas. And yeah, there's a fellow named Kenneth Miller who's a professor of biology at Brown University, a very smart guy. But he says, yeah, I can reduce a mousetrap and take away the hammer and use it as a tie clip for my tie. And I was debating him once at the American Museum of Natural History in New York City back in 2003 or 4 or something. And I said to the audience, I said, men, do any of you have tie clips on that look like this?
Oh, too bad.
And I said, if you did, you probably couldn't get a date to come with you. And so that mutation will disappear. But the interesting thing is that, more seriously, if you're going to make a tie clip, you're not going to have it in the shape of something that you want to get to be a mousetrap. He is using these things for purposes that he wants to lead to his goal, which is the mousetrap, to show that there is a pathway. But a real tie clip would look a lot more elegant than that. And therefore, even in Darwinian theory, that mousetrap tie clip would experience selective pressure to change to just that function that it was doing right now, to be a better tie clip, not to be a good precursor to be a mousetrap later on. Darwin's mechanism doesn't care about the future. It only cares about right now. So if it can get rid of something or change something to better fit its purpose right now, it'll do that in a flash with no thought at all to what could have happened in the future.
So if we add a caveat to the definition of irreducible complexity, maybe correct me if I'm wrong, we would say a system is irreducibly complex to the extent that if you remove one part, it doesn't function in the context of its own very intended function.
Right.
So if we can just remove the hammer from a mousetrap and use it as a paperweight or whatever, a tie clip, it is not anymore a mousetrap. So obviously we have lost the functionality of a mousetrap. So this actually proves nothing at all. Isn't this a fair statement?
That's exactly right. The definition is that if you remove a part, the function of the system is gone. It doesn't say that you can't, anything can be used as a paperweight, anything that has mass. So essentially they're trying to explain away what any child could see as a problem for making a mousetrap, in a Darwinian fashion. But hey, they got to say something.
But I want also to ask you something else, because I have seen some of those arguments, and I'm really pissed off that really the masses are they're deceived by these examples. So let's just assume, okay, that somebody has a very bad taste that he is using a dysfunctional mousetrap as a tie clip. Let's say. Okay. Let's say that somebody that this guy arranged with 20 people in the theater or in the museum to come with mousetrap tie clips. Okay. And that he used a conspiracy to make it the fashion of the day. Let's just assume it for the argument of the discussion. Wouldn't it be a condition, according to the Darwinian paradigm, that we would find an ancestral form of the mousetrap that is a tie clip on its lineage, on its very lineage. Otherwise, whether people are using mousetraps as tie clips or not is completely irrelevant.
Yeah, that's interesting. I don't know. Yeah, I guess if you were Darwinist, you would predict that you might find a mouse or a tie clip kind of structure in the distant past. But number one, if you didn't, that wouldn't matter anyway, because you would just say Darwin's theory, of course, we just haven't found the right pathway. Number two, if you did find that, number one, it wouldn't explain where the tie clip came from in this peculiar arrangement. And number two, it still wouldn't tell you how to get from a tie clip to a mousetrap by an undirected process, which each of those steps where Kenneth Miller and other folks, 25 years ago, I proposed this mousetrap as an example of irreducible complexity and a cottage industry of trying to build mousetraps from simpler starting materials has arisen on the internet. And last time I checked, nobody has been able to do that, at least to a skeptic like myself. Each of the steps in any of these scenarios, if you have your eye watching for it, you'll see the intelligence of the person proposing this pathway entering into how to shape and how to put things together. It's not done by random processes. The humans trying to make that argument are guiding it along the path that they want it to go. So even if you did see something resembling the parts of a mousetrap in the past, that still doesn't tell you, number one, how they got there, and number two, how they could be assembled into a functioning mousetrap.
All right, I will refer back to this point again later in the discussion. But let's first ask, if irreducible complexity is true, and I definitely do believe it's true, how does it debunk neo-Darwinism or unguided evolution in general? What's the killer point there?
Okay, well, it goes back to Darwin himself. In The Origin of Species, he said that if it was shown that some organ existed which could not possibly have been formed by numerous successive slight modifications, that his theory would break down. Mm-hmm. And he said he didn't see any examples, but I think we've got some today. And in that, Darwin was insisting that evolution had to be gradual. You had to change things a little tiny bit by small steps over long periods of time, so that selection could choose one and then choose a successor and the next one and the next one, because he knew that if things improved in leaps over big distances in short time, then it would look very suspiciously as if randomness were not the driving force. It would look like something else was involved. So the problem with irreducible complexity is that they can't be put together by these numerous successive slight modifications, of Darwin. Essentially, with an irreducibly complex system, it's got to all be put together before it functions at all. And so the problem is that natural selection doesn't have anything to select until pretty much the whole thing is there. Or in the case of the tie clip, it has something which you'll select to be something else rather than what you're trying to build. And so if you have to put everything together to make a complex system before natural selection can kick in, then Darwin's theory is essentially not doing any work. It's just waiting for complex things to be there, and then, it'll select it if nobody else has it, but it's not responsible for explaining how it was built up over time. And
Amazing. Yeah.
all other theories, unguided theories, have the same problem.
All right. All right. So we would say that if something which is irreducibly complex, if some of the parts come by chance, then according to the theory itself, natural selection should throw the whole incomplete thing away anyway, because it has no function without
Exactly.
its complete parts and its complete proper
Exactly.
arrangement also. Yeah.
So that is essentially saying that chance, put together a complex machine. There's no difference from that. But it's interesting. I should point out that irreducible complexity has another very relevant point, other than just being a problem for Darwin. It's also a strong pointer to intelligent design, because as we discussed earlier in the podcast, we detect intelligent design by the purposeful arrangement of parts. And so if you have a system in which the parts are arranged so it can do something like a mousetrap or a flagellum or so on, then that is strong positive evidence for purposeful design. Turns out that's how we recognize design. We don't read minds or anything, so we recognize design in the purposeful arrangement of parts.
Great, because this is not only a purposeful arrangement of parts, but it's also a purpose arrangement of parts that are intricately picked to the extent that if one is missing, it's just not going to work.
That's right. So that's a problem for Darwin's gradual theory. Yeah.
All right. So now neo-Darwinists typically pick examples to counter irreducible complexity. Not necessarily good examples, in my opinion. So what is the criteria to determine if a system is irreducibly complex? Let me expand on that. They would just come to you and say, okay, look at this system. If we take that one part, it will still work. And so irreducibly complexity, and then they would go irreducible complexity is not a thing. Mm-hmm. So how is this argument flawed in your opinion, and what's the proper response?
Well, you have to pay attention to the details of the system, and
I'll give one example just for the audience. I think one of the examples that you previously gave was the blood clotting cascade of reactions.
Yes.
And I don't remember which scientist was it that I saw he was making the presentation. And he said, well, there are other blood clotting mechanisms in other organisms that does not have this or that step. So it seems that the blood clotting mechanism is not irreducibly complex in that or other sense. And they would also give some other examples. So what's the proper response for that if this is the case?
Okay, well, you have to pay attention to the system itself. So one kind of rhetorical problem that intelligent design has versus Darwinists is that Darwinists want you to think evolution is easy. It's not a problem. And so they don't point to any problems that it would have to overcome. On the other hand, intelligent design folks want to tell you that evolution is extremely difficult or impossible, but in order to do so, have to point out all these details that nobody wants to listen to. So, so it's a problem. But let me first use an example of not a mouse trap, but a rat trap. I have a rat trap on the shelf in my office. And it's got all the parts that a mouse trap has. They're a little sturdier, but it's also got two springs to generate more force because of the larger rat. And if you take one of those springs away, it'll still work after a fashion. Maybe it'll catch some weaker rat. Okay. So if a Darwinist came up and says, look, I can take away this extra spring and it still works, I say, yeah, well, that's because that's not the irreducible part of the machine. And other people have said, well, you would need cheese or something. And if you take away the cheese, maybe it would still work. In the case of the blood clotting system, the blood clotting cascade has a couple of different options, kind of like the rat trap. It can be activated by one branch or something called the internal, or the other is the external branch. And I discussed this in Darwin's Black Box. They both kind of funnel into a pathway of clotting that uses the same 4 or 5 proteins at the end before a blood clot is formed. And I said, well, leaving beside all that stuff after the fork in the pathway, if you look at this, you've got problems. You can't build this up gradually. And astoundingly, well, maybe not astoundingly, the first criticisms I heard were from Ken Miller, who says, well, look, after the fork in the pathway, you can take away one of the proteins and that works. I said, yeah, but I explicitly said, after the fork in the pathway. So one gets a little discouraged because you worry that you're not discussing this in good faith. But interestingly, the Kenneth Miller doesn't work on the blood clotting cascade. The research was done by a man named Russell Doolittle, among others. Mm-hmm. He was a prominent biochemist. He died a couple of years ago. And he wrote a book called The Evolution of the Blood Clotting Cascade, I think it was called, in which he just noticed that there were proteins that were similar to each other in earlier, simpler organisms. And then there were proteins with similar sequences in higher vertebrates and so on. And he says, well, there it is, but as I try to emphasize, just pointing out similarities between things doesn't say at all how they developed or how they may have transformed into something more complicated. It's kind of like, that's not a good analogy. So that's one big problem. Another big problem in biochemistry is that people will call proteins by the same name in different organisms. Say there's a molecule called fibrinogen, and it's the precursor of the material that makes a blood clot, the kind of the webbing, the ropes that make a blood clot. Mm-hmm. And it has to be activated to do that. And there's a little piece you have to cut out, and then it kind of self-assembles into this. But fibrinogen in people is not the same as fibrinogen in fish. And fibrinogen in fish isn't the same as other creatures too. And so sometimes proteins do a couple of jobs in one species where they are split up and do other things in other species too. So I guess the long and the short is you have to look at the details of this. And when you do, in my completely unbiased opinion, you see that it doesn't get one. It doesn't pose any problem to my arguments.
So in summary, let's say that if you propose a system that is irreducibly complex, it doesn't need to be the whole. There is a core part of this. There is a hardcore part of the system that if it remains irreducibly complex, the argument stands, irrespective of all the accessories, let's say. Because at the end of the day, you will need to explain how the core came into place, right, and whether there are 5 or 6 or 7 or 8 or 9 versions of the same core. So let's say that the Chinese and the Americans and the Japanese all arrived to inventing the mobile phone, and each one has a different core technology. Like at some time in the US, there was CDMA, other places there was GSM, etc., and different things. It also does not change the argument, because that there are different configurations of how to capture mice in a box or under a hammer or in a hole or whatever. Each of them can be irreducibly complex. And you're even actually making the argument more difficult, because now we need to explain how 3 or 4 different variants come into play that are not interchangeable. So,
yeah, that's right. And it's, let me assure you, it's surprisingly difficult to try to change even things that look very similar to each other into each other by an unintelligent Darwinian type process. Because one problem when you're thinking about things like this is you say, okay, well, we've got this and I want to go to here. So how can I do it? But it turns out that random mutation says, I need to survive. What if I get rid of this? I'll have the extra energy to divert to something else. And it's not thinking of doing something else. And turns out the processes that could degrade something are a lot faster than processes that would make something. So I think the best insight into what happens in this is given by Richard Lenski's experiments on bacteria that we discussed a little while ago. The E. coli that he grew in his lab had a flagellum, and one of the first mutations to occur was one that shut down the flagellum so that it wouldn't work anymore. So, if your machinery is one of the first things to be thrown overboard to help the ship stay afloat, then it's not going to develop into something more complex.
And in this case, that bacteria was actually spoon-fed. So if you really put it later in the wild, it will really have a hard time surviving now, right?
Yes, that's exactly right. It would not do well. It's kind of like having a French poodle, in the wild.
Yeah. All right. So great insight. So next, I would like to ask you this. You've talked about cells having abilities to compensate loss of one function from one pathway to another pathway, and evolutionists or Darwinists typically like to use this in a certain way to say, hey, look, things are not really irreducibly complex, because when life finds ways, and then they would extrapolate that those ways are because of the random mutations that's creating different variations, etc. And then we find experiments that show that this is not what happens. As you say, always on the molecular level, there is where the trick is. So what's your insight? Is it when we find this resilience of life, this amazing resilience of life, that, for example, that we build all those antibiotics and it turns out that those bacteria have always this plasmid up their sleeve, and they throw a wild card and suddenly okay, they had it figured out like billions of years ago, and obviously not because they went to school or to university or they had a crystal ball or anything. What does this say about the design of life or about the structure of life, in your opinion? Is this something that is with or against intelligent design in general?
I would say it's hard to say whether it's with or against intelligent design, but I think it's pretty easy to say that it's against Darwinism, because if Darwinism does not sit well with redundant functions, that if you have engineers know all about redundant functions, they have backup systems in case something fails, but that requires intelligence and foresight and so on, expecting what might go wrong. Whereas Darwinism is, just looks at the moment. It can't plan for the future. So if an organism has one, say, metabolic pathway that can do something, but if it suffers a big mutation, well, there's another that could do the same thing pretty well. That it should not be predicted by Darwinism. Of course, when it's discovered, then turns out that Darwinists say they do predict it, but these
in hindsight, always in hindsight.
Yeah, hindsight is 100%. But yeah, that, and there are a lot of really strange examples of genes that seem to be critical, but if you get rid of them, it doesn't matter. I remember being struck, I guess it was 20 years or so ago. There was a paper in Nature, which is, of course, the most prestigious science journal in the world, and it had this wonderful title. It was titled "Mice Without Myoglobin." Now, myoglobin is a little protein in your muscles that binds oxygen, and the purpose of it was thought to be that it will have a reservoir of oxygen for when you go out jogging and you need extra energy, and the oxygen can metabolize foodstuffs and give you energy. Well, these researchers took a mouse embryo and somehow knocked out or destroyed the gene that made myoglobin. And when the mice developed, they grew up and they didn't care that they didn't have myoglobin. They lived, they behaved the same way. They even could run on a little spinning wheel and exercise.
Amazing. How did they do it?
I don't know. It's still unknown. But so the question is, what's the function of myoglobin if you can get rid of it without any harm? So there are a lot of systems in the cell, and I'll say their purpose is hazy. They do something specific, like bind oxygen for myoglobin, or maybe catalyze a particular chemical reaction over here. But it's real hard sometimes to say exactly what they are for, what in terms of the survival of the organism. So I think there's not enough data. We don't know enough to reach any big conclusions about design from the redundancy of functions or the apparent dispensability of functions like myoglobin. You can suspect that, well, these things are probably doing something of which we are unaware, or they're part of systems that we don't know about, or have some future use for all I know. But we can, so we can't, I don't think we can conclude too much from that. But the nice thing about intelligent design is that you can conclude it based on the structures of many things, just by looking at them. Just like with the mousetrap or Mount Rushmore, you don't have to investigate it. You just look and say, oh, that was purposefully arranged. And there are so many things like that in the cell that you can afford to just keep wondering about what other mysteries in the cell will be determined in the future.
It just came to my mind while you were talking. For example, I'm designing a submarine. Okay, and I would put in the submarine a radar system that works on electromagnetics, but I also have a sonar, right? And then somebody says, well, this design is stupid. I can just disable the sonar and the submarine will just work. And then we go into a battle where the enemies are using electromagnetic jamming, and then now we do need the sonar. Otherwise, we'll really be hit with a torpedo, and we'll not even know what hit us. And I think if somebody who doesn't really know that there is something called electromagnetic jamming that can actually jam a radar, or that there is something called stealth technology, and that we have this other kind of electromagnetic detector that will actually work even with stealth submarines, that I think what you're saying is that what we don't know is so huge, given the complexity of life, that we don't even know why things would just keep on working while we turn things off, and why there's all those so many things. And it strikes me the fact that life has been on this planet for like 4 billion years, and that it has gone through so much. So we really don't know what's really important for life to arrive to where we are today, because at some points there weren't so much oxygen, the temperatures were different, the acidity of the environment was different. There was so much different. But do you agree that it's unfair for us, given our very primitive knowledge, given the complexity of life, to make those assertions? And it's not really very scientific for Darwinists to make those assertions if we only know so little.
Yeah, and that's exactly right. And if you look through the history of biology, it's been very, very frequently the case that when people didn't know what something did, they would conclude it had no function. So back in the early 20th century, people didn't know what DNA did, and so they thought it was a simple kind of structural molecule, not very important. And that's that has occurred at all sorts of levels. So there might be, the cell almost certainly is doing many other things of which we are utterly unaware. But we should be careful to not kind of flip to the opposite end, because we do know some things. We do know some things of how it works. We know how the sonar works, and we would know how the radar works, and we would be able to tell just from them that they were designed, because they
Absolutely.
have these parts. If even we don't know the other one or what the rest of the submarine is doing, we know that they were designed. So if you look at the blood clotting cascade, you look at the flagellum, intracellular transport, and so on, you say, hey, this is purposely designed. There might be lots of other stuff in the cell that we don't know about, but we do know that much of it is required purposeful design.
Yeah, I just I want to ask you this other question, because I also see it in the chat, and we had it later, but I'll push it earlier. So it's about, I know that you have answered to this many times, this type 3 security system stuff. So some of the irreducible complex systems seem to have other systems that look like them, that do other functions. Example, a flagellum looks like a type 3 security system. Obviously, the flagellum has more parts in it. So some twist this to attack irreducible complexity. I think we have already talked about it in terms of analogy by talking about the mousetrap and the tie clip. But just give us some hint about this, because it keeps turning up, and it is one of the things that really strike me bad, that you have done all of those responses, all of those rebuttals to all your critics, and I don't know why people don't know about them. It's really unfair. So if you can just give us this one example, and it will be great.
Okay, yeah. The flagellum, as we said, is this outboard motor type thing, spins around and around. And it's really remarkable because, unlike outboard motors in our world, which are put together by people or machines made by people, the flagellum has to assemble itself. These complex systems have to get put together. And it's really very intricate. And it turns out that the motor of the flagellum, the thing that turns it, is on the inside of the cell and in the cell membrane. And it turns out there's a hole in the middle. It's kind of like a tube, and the outer parts of the motor and the flagellum are sent up through the tube and placed on the ends to build it. And here comes the flagellum, and that it has to be very carefully done, and there are all sorts of auxiliary proteins that help do it. So that's how it's built. Now, in other bacteria, or in some have both, there's a structure called the type 3 secretory system, or TTSS, and it turns out that it has some proteins, not all of them. It has some proteins that resemble proteins found in the flagellum, in that it has a motor and it's got a tubular structure and it has kind of a pointy thing coming out the top. And it turns out, though, that it's not used as a rotary motor, it's used as an injection device, kind of a syringe, where it can push proteins, poisonous proteins, into prey bacteria. Maybe ones that'll break down its DNA or something and kill it, and then the predator cell can absorb all of those materials. So some people said, hey, this has similar stuff to the flagellum. Maybe this type 3 secretory system arose first. It's kind of a simpler system. And then it went on to develop into a flagellum, a rotary motor. And it's interesting because, well, I respond generally by saying, number one, that folks who say this don't explain where the type 3 secretory system came from. It's just there. That's where they start from. They don't explain how it could have given rise to a rotary flagellum. It's just the endpoint. And so they haven't explained anything except to note that, gee, these things look kind of similar to those. We mentioned earlier that natural selection would kind of fit a system to its current role. Now, the role of a type 3 secretory system is as a needle. There is nothing in it that is tending towards a rotary propeller to drive a bacterium through the liquid, and so there's nothing that would pre-adapt it or try to push it that way. And again, that has been unexplained by Darwinists. So at the end, you're left with the intermediate, the type 3 secretory system, which is unexplained, the flagellum, which is unexplained, nothing about how you got there or how you got in the steps between, but just the assertion that because this kind of looks like that, it must have arisen from that. And it turns out that currently, or as recently as 10 years ago, it was established that the type 3 secretory system is younger than the flagellum. That is, it seems that the more complex apparatus arose first, and then maybe somehow degraded or something, and now you can use parts of the flagellum as this injection apparatus, and that might be interesting. It might be.
That as big a blow as that is, they still use it. They still use the example, and it's really astonishing.
Yeah, I agree. Much of the battle is not over truth; it's about dominance, I think. Yeah. But I should say that might be true. It might be true that the flagellum came first and decayed in some places to this other type 3 secretory system, but it's no Darwinian process that says, oh, we have this unexplained, extremely complex system, and it gives rise to a simpler system. That's completely compatible with intelligent design, but not so much with Darwinism.
Yeah, but I would even think with you. I'll just take this one step further. Number one, I don't believe as an engineer that a flagellum degraded into a syringe. Okay, it completely doesn't make any sense. But let's assume, just for the sake of the argument, that this is what happened. All right. So let's assume that somebody, after your debate with Ken Miller, says, "Wow, this can be the fashion of 2023, and we will actually have tie clips that look like mousetraps because we want to tease these ID guys." All right. And then they start producing those tie clips that look like mousetraps. Doesn't that on its own require an intelligence? If somebody would say, wow, this hammer can also be with a little modification. If we make it like shorter or longer from that place, would be used like a pick axe or something. Okay, isn't that also a work of an intelligence that will transform one irreducibly complex system to another irreducibly complex system? In other terms, let me tell you, if somebody looks into the design of a radio and it inspires him that he can make a new device that would pick a signal that is actually a video signal and call it a TV, okay, doesn't that, or somebody looks, if let's assume that the TV was invented first, and then somebody says, wow, we can actually reduce it and make something new, which is called a radio that will only pick audio because people who are driving shouldn't be distracted by images, for example. Doesn't that on its own require intelligence? And would it not be absurd that somebody says, well, there are those similar components between a TV and a radio, and then the TV must be a product of an evolved radio. And then he insists, even after finding that the radio came into the history of technology after the TV, which is not the case, but it is the case in the case of the flagellum and the type 3 security system. So don't you agree that even this degradation would require intelligence on its own, because there are...
Yeah. Oh, sure. And I'm sure engineers would know this a lot better than biochemists. But yeah, if you have another complex system that somehow seems derivative from the other, you've got to rearrange wires and you've got to put things in different places and all sorts of stuff, which is, to say the least, unlikely to occur by a random process.
So I mean, if I give you a flagellum and I tell you, as a biochemist, can you drive through mutation a secretory system that actually works? And it gets its feed of those poisonous proteins, and there is the whole integrated system is sitting behind it, and the bacteria understands that this is now a way to attack prey. Okay, does this lend itself to be the process of just random mutations and natural selection? Can you actually do it in the lab even?
You could probably try. I would bet against it succeeding. But yeah, I mean, you could do a Richard Lenski type experiment and again, select for a predatory function on some other bacteria and see what random mutation is likely to do. And that's the best thing. I want Darwinists to do experiments like Lenski's experiments, because it's always been the case that whenever they do, then we see that it explains less and less and less than we had thought before. So it'd be great if somebody would do that.
Yeah, so I think for the audience and for people who actually still talk about the type 3 secretory system and flagellum, I think that was a great answer. Now, are we okay to keep going, or
Sure,
you're
sure.
all right?
Yeah.
Okay. So one other argument. Again, I like to tell the audience that we will answer questions still at the end, so you can keep pushing questions if you like. So one of the things that I see naturalists push on the table is that they say that co-option can actually solve the irreducible complexity question. Can you first explain to us what co-option is, and does it or not defy irreducible complexity as an argument? And if there are actually real examples on the ground of co-option that we can relate to.
Well, co-option is a word that means a pre-existing system which was doing something else was somehow pulled aside or duplicated, because genomes can duplicate genes and stuff, and reassigned to do a different function. It was readjusted somehow. That's always a little fuzzy. And now it does a different a different function. I remember reading about co-option in Stephen Jay Gould's, one of Stephen Jay Gould's writing. And his example was that in East Africa, I think we was talking about Kenya or something, that people would take used tires and cut out pieces and use them as sandals. They put little latches or ties on them and use them as sandals. And, oh, okay. He says, well, it was a tire, now it's a sandal. It's been co-opted. And I was very, very unimpressed with his example, because there's no complexity involved. All you've got is this surface. And even so, you've got intelligent agents that are picking these up.
And sandals happen to be irreducibly complex on their own, because if you don't have the sole, if you don't have the band, if you don't have that or this or that, it will not work as a sandal. And it became a sandal because an intelligent agent actually used material, made a purposeful arrangement of parts. So
right. Even things that appear simple, you know, when you're not thinking about it, when you try to actually make it, it becomes a lot more complicated. But in the cell, one classic example of co-option is, well, not in the cell, but in an organism, our lungs, people have speculated that they are reassigned swim bladders that fish first had. Fish, you know, hold air in their swim bladder, which helps them float at the right level for their lifestyle. We have lungs, and some reason people think that, well, they both have air in them, so maybe one turned into the other.
Wow. Yeah, this one really knocks me out when I hear it, because
but nobody goes, you know, through the details and said, well, this is why this would be better and this would be better. And so it's essentially, you know, a just so story that Darwinists are famous for. And there are other examples too of various proteins that have similar amino acid sequences. For example, there's a protein called serine, not serine, a serine protease. And one serine protease is a protein called trypsin. And trypsin is what's called a digestive enzyme, because it can hydrolyze or can break proteins down into small parts and in your stomach, and then the parts, the amino acids, can be absorbed and reused. It turns out that a number of the protein factors in the blood clotting cascade are also serine proteases. That is, they have a family resemblance to trypsin. So some people say, well, there you have it. That explains the blood clotting cascade. You know, it was just co-opted because, you know, there was trypsin.
Wow.
But again, we're seeing a similar pattern here, that if something looks similar but has a different function, and somebody sees something else over here that sort of kind of resembles it, has a different function, they take that as sufficient evidence, not just that they're related, but that they were developed by random changes and natural selection. And that's an utter non sequitur. It just, you know, there's no evidence that one could give rise to the other. And all of the problems with gradually developing a blood clotting cascade that I discussed in Darwin's Black Box don't depend on where the original protein came from. It's the control of the clotting cascade and the targeting of the different proteins and so on. So Darwinists have a lot of kind of catchphrases that they apply to different problems, but they're just labels. They're not explanations, or they don't explain anything. They just cover their ignorance with a word.
So it's like, again, the existence of some of the integrated circuits and electric components in the FM radio is not an explanation for the existence of a TV.
Right.
Yeah,
Exactly.
just because there are some shared components doesn't mean that this
Uh-huh.
explains how a TV exists, because the
Yeah.
radio and the TV need the designer, and whether they depend on similar components or not is completely circumstantial. It is not an answer to how a TV came into existence.
Exactly. Yeah, that's exactly correct.
But is there real examples that can be proven on the ground for co-option in action? Or it's just one more of those, name tags that just gets, filling whole fillers, let's say.
Well, I think that there are examples of single-celled organisms where bacteria lives within a larger eukaryotic single-celled organisms. What exactly that has to do with co-option, it's not clear. And you really have to know, again, the level, the details at the molecular level to know what's going on and whether it was possible by a Darwinian process. The classic example of, well, co-option and symbiosis is the mitochondrion. There's in eukaryotic cells that are everything except bacteria, they have a nucleus and they also have this little subcellular compartment called a mitochondria, which is actually responsible for producing much of the power of the cell. And a lady named
actually I had a question about symbiosis.
Okay.
So I say that some appeal because I think they actually use symbiosis as a different category rather than co-option. So let's talk about symbiosis. So some appeal to symbiosis to explain complexity. What's your comment on that? And is there real examples to that has unequivocal evidence? And since you are already mentioning the example of the mitochondria, and I think they postulate that it came from cyanobacteria or something. I'm not sure. Or maybe it's the chloroplast that is so. But okay, let's take symbiosis. What is symbiosis or endosymbiosis in this case in concept?
Well, endosymbiosis means that two organisms live together and to the benefit of both. And endosymbiosis means that on the cellular level, that one cell lives inside the other, and somehow that benefits both of them. And the classic example is the mitochondrion. And it's thought that the mitochondrial DNA has some features that are reminiscent of bacterial DNA. So people speculated that maybe in the distant past, a bacterium invaded and got inside a eukaryotic, more complex cell and just survived. It somehow wasn't digested or killed off. And that over time, the bacteria, which had the ability to make energy more efficiently, say, than the eukaryote, specialized in that activity, and the eukaryote specialized in other things and would supply the mitochondrion with whatever it needed, and together they lived happily ever after. But
that was a real good one. But okay.
Okay. You can raise particular objections to the scenario, such as how would proteins know to get into the bacterium in this situation, and other things too. But in my view, the basic problem, or the basic reason this doesn't explain anything, is that you already had the bacterium and you already had the eukaryotic cell, and they already had all of the functions that they're ascribing to the new system, the cell with a mitochondrion in it. So you're not
actually. I think their argument is much worse, because I remember that they say that it was one bacterium that engulfed another bacterium, and that eventually gave rise to the eukaryote. And I don't think that. So they don't even start from the eukaryote at all.
Yeah.
So it makes it even much worse.
Yeah. Well, there's this. People have proposed a series of such things.
Okay. All right.
So,
So if they start from the eukaryote, how do they explain it in the first place?
how do they explain the eukaryote or the?
Yeah. How do you explain that the eukaryote is there in the first place without mitochondria at all? So it's what is this? A handicapped eukaryote or what?
Well, the way they explain it is vaguely.
Okay.
They just kind of wave their hands and say they got together, and the first one specialized in being the nucleus, in holding on to the DNA of the cell. But an analogy I like to make with symbiosis is, suppose you had an automobile, you were driving an automobile, and it didn't have air conditioning, and it was a hot day, and you were driving along, and a room air conditioner from an apartment building fell into your car.
Okay.
And somehow it's still running. Now you've got a car with an air conditioner. Maybe it gets fixed up over time, but you didn't explain where the air conditioner came from or the car came from. You just say they kind of fell into each other. So I don't see symbiosis as much of an explanation either. It might be, true as a fact about the history of life, but it's not evidence for Darwinian processes. It would certainly have to be intelligently guided to amount to anything.
I remember I was challenged by an evolutionary biologist on that, and I happened to be ready with it, and I was struck by the fact that I think that a huge amount of the proteins that build the mitochondria actually are from genes that are in the nucleus.
Yes.
So I can't see, and I think the ratio was 90-something%. So this, if this miserable cell used to be independent, I cannot envision, because correct me if I'm wrong, losing like something in the order of 1 or 2% of your DNA by mutation will kill an organism, right? So if an organism,
unless you pick exactly which pieces you want to pull out, probably.
Yeah, but then if you lose like 90%, is there a biochemical process that will cause this unbelievable synchronicity that once you lose a gene from here, it will just miraculously appear in the nucleus of the eukaryote to the extent that is this possible in the lab or something that you know?
Well, people, don't look into it too closely, but they assume that initially the bacteria that became the mitochondrion had all the genes it needed, but then over time, they say, it would transfer, or some of its genes would get transferred to the nucleus so that it could it specialize more closely in producing energy. Exactly how that happens is unknown, but we know that DNA can get absorbed by cells, and genes could get integrated into DNA. So there's, it's hard to put your finger on exactly, what would be difficult, but it requires some pretty amazing steps.
So is it like a trend that no matter what is the proposition, it occurs to me, but I really want to get your catch on this, that no matter what is the mechanism that's proposed by the Darwinists or the unguided evolution proponents, that once you go into the details, you find a big gap. There is always no path. I see this like a trend for almost every single one of them that we have gone through, that once you just look under the hood and look for the mechanisms or how things will really move from A to B, it's like a handful of miracles or something.
Yes, absolutely. Yeah, that's right. So the one reason Darwinists and their theories survive is that they don't do experiments to try to test these assertions, or if they think something is reasonable, they just propose it and everybody nods their head and they move along. So they don't have very demanding criteria for trying to decide what is correct or not.
Yeah, and actually, because we mentioned mitochondria, I think they posit the same story for the chloroplasts, and they say that the chloroplasts also had endosymbiotic origins. And I was having this interview with Dr. Günther Beckley, and he told me that photosynthesis just appeared in the fossil record fully formed at a point of time, just few million years, or just at the very early history of life. And obviously photosynthesis looks like, does it qualify as an irreducibly complex system in your opinion, or does it have an irreducibly complex core? And what does this say? What does this say about intelligent design and irreducible complexity in general?
Well, I think it means in general that if you're going to if you need a system to perform a function in order to live or to get to the next step, it has to be designed. If a mousetrap has to be designed, everything does. The fact that systems appear really pretty suddenly, or subsystems, and both in the organismal fossil record and in studies of genes and in chromosomes, indicates that something is coordinating. Something is coordinating changes in life, and it's not accidents that are coordinating it. It's interesting these days that as it's only been the past 10 years or so that science has gained the ability to sequence DNA, the complete genomes of DNA of whole organisms, pretty easily and rapidly. And what a lot of genome studies are showing is that in one branch of life, say yeasts, the number of genes, particular genes, would decline, and then after a while, for some reason, there would be sort of a jump in the genetic information of these things, and then it would start to decline. And to somebody who's not wedded to Darwinism, that looks like a lot like teleology right there. That's a decent marker for it. But of course, since it's complex, you have to work out a lot of the details. But it doesn't.
So this is like what explains saltations on the fossil record, on the molecular record, that there are those sudden infusions of information that just come there, and there's a new arrival on the theater. That's right. Be it an eye or photosynthesis or whatever.
Yeah, whatever is appropriate. That's correct. It's really completely different from Darwin's idea of gradual change.
So actually it takes us to our next question, which is what you proposed in your, I think it's your last book, which is Darwin Devolves. That the general trend is that you start with something better, and actually mutation makes it, breaks it, right? So you mentioned de-evolution. That's why I think you called the book Darwin Devolves. Right. So you mentioned de-evolution by loss of function, rather than evolution by adding novelty. So the Darwinian mechanism actually causes loss of function. So how does this relate to irreducible complexity? And if species come from a degradation of ancestors, like I think you gave the example of dogs and wolves, and of the polar bear versus the traditional brown bear, then when and how does the gradient become positive? I think this is what you've just said.
Yes.
It just becomes.
We have no explanation for it. There are certainly no details or mechanisms. But as far as devolution, just for listeners, the interesting thing from science's new ability to sequence DNA is that you can actually track down the changes, the mutations that happen, which are beneficial and which give an organism an advantage. Back 20 years or so ago, you couldn't do that just because the machinery wasn't available. And one really interesting thing is that most of the mutations that are helpful, that are beneficial to an organism, are ones that break or degrade pre-existing genes. And I talked about this in Darwin Devolves. I started out with the example of the polar bear. Polar bears are, really tough animals, but it turns out that folks who were interested in evolution sequenced the entire genome of the brown bear, grizzly bear, and the entire genome of the polar bear. And polar bears are thought to be descended from brown bears. And so they wanted to directly compare to see what happened to make this really nifty new species. And it turns out that of the top 17 genes that were mutated, which helped the polar bear thrive, 3/4 of them were degradative mutations that either broke a gene or made it work less efficiently than it did before. And the first one is easy to understand. It's the change in the loss of the ability to make pigment in the fur of the polar bear. That matches it to its surroundings nicely, but it's a match that is brought about by breaking something that was already there. And it turns out that it's real easy to break a gene because genes are very complex and they're like computers or something. It's a whole lot easier to break your computer if you hit it with a hammer than to make an improvement to it. Nonetheless, I guess the interesting and new point is that many times you've got 10,000 different genes. Turns out maybe you'll survive better in a particular niche, in a particular environment, if you get rid of one of those genes. If you are on a sinking ship and you have to make it to port, your best chance to make it there is to take complex, heavy machinery and throw it overboard. And that might help you survive, but it doesn't explain how that machinery got there in the first place. And the examples that Darwinists have been using forever as proof of the power of Darwin's mechanism have turned out to follow this line of circumstances. That, for example, dog breeds. Darwinists have always pointed to dog breeds because dogs come in all sorts of different varieties, different breeds with very different sizes and shapes and so on. It turns out that almost all of those characteristics are explained by breaking some element of the breed's genome. If you want a small dog, you can break the growth hormone. If you want a large dog, you can break the control elements that tell the cells when to stop making growth hormone. If you want a dog with a very short muzzle, you can break a gene involved in facial development. And interestingly, if in the wild it would be beneficial for a wolf or something like it to be smaller or to have lighter fur or something, then it would be just as easy for it or easier to break a gene than to come up with a whole new way of making something. And the faster the mutation arrives, which can affect the selection pressure, that's the mutation that's going to kind of dominate the population. So it's once you see it in retrospect, you see, well, of course,
yeah.
But still, it's only in retrospect. But Darwin's theory now has two completely independent problems. The one is irreducible complexity, it just can't build things that have multiple parts that are purposely or that need to match each other in order to work. And that's a separate problem from the problem of devolution, that it's oftentimes easiest to adapt to an environment by breaking something you already have rather than making something new. So I myself think that is those separate problems really put Darwin's theory well beyond credibility and
yeah. So actually, it's great that you mentioned that, because I have seen some people making a mix between your Darwin Devolves book and Darwin's Black Box, and they think that Darwin Devolves is coming in support to Darwin's Black Box. While what you're saying now is that they're actually two independent arguments. Each of them, at its own right, is a big category of issues that Darwinism has to deal with, and that unguided evolution cannot explain irreducible complexity, and it cannot also explain the reverse gradient of information. If only what happens is devolution by mutation, then where does the sudden infusion, not just infusions, but huge sudden infusions of information come from? Did I get that right?
Yeah, that's exactly right. So it's kind of like, if you have to run a path that has hurdles on it, you got a hurdle there, hurdle here, and then all of a sudden, also there's a gorge which you have to jump to. They're independent problems, and either of which would be sufficient to rule out Darwin's mechanism as an explanation for complicated systems.
All right. So you've mentioned before extended evolutionary modern synthesis. Sometimes it's abbreviated EES, sometimes EEMS, whatever. So it's just an acronym. But under that, you mentioned something like natural genetic engineering. And actually, the term pisses me off because I don't think Natural Things know anything about engineering anyway. Set aside genetic one. But what is it? What does this term mean anyway?
Well, it was coined by a man named James Shapiro, who was a microbiologist at the University of Chicago, and he's another skeptic of Darwin, but he doesn't like intelligent design. And he said, well, in our laboratories, biochemists, molecular biologists oftentimes use the enzymes from cells in order to rearrange DNA and clone genes and other things. We take the enzymes that make DNA, ones that cut DNA, ones that modify it, and so on, and we do genetic engineering. Mm-hmm. And he says that's kind of like back in the day when Darwin was thinking. Darwin said, pigeon breeders, they select the qualities of the pigeons that they want to have in their future line. And he said that probably nature does the same thing. So he called that natural selection. And James Shapiro says, well, maybe we have natural genetic engineering. So nature genetically engineers too.
But is there a mechanism for it? Is it just just another name tag?
Well, no. I mean, you talk about these transposons and so on. There are little elements of DNA that have information in them already to code for enzymes that will cut them out of their current place and put them in to a different place in the DNA. And there are other enzymes which will chemically modify DNA with a methyl group in one reaction called epigenetics, and they can make the DNA more active or less active. And so you can say to yourself, well, this is genetic engineering, and that it's turning genes on and off or switching them around. And if it's occurring just in the context of the cell, well, you can call that natural genetic engineering. The interesting question to me is, well, okay, number one, it doesn't say where all these tools came from, where the enzymes that engineer the cell came from in the first place. So already to get there, you have passed over a whole lot of development of a cell. But where is the evidence that these things can do anything when left on their own, rather than in the direction of scientists and laboratories? For example, again, going back to the swell work of Richard Lenski, we didn't see any fancy genetic engineering in the E. coli cells in his very long-term experiment. We just saw degradation of stuff.
Or using what's already there to complete a pathway for citrate by this crazy transposon jump, which looks to be an intentional reactive adaptive response, actually.
Yeah. Well, yeah, perhaps so. But even beyond that, though, there's nothing much going on. And if these elements go beyond their normal activities, say this enzyme is supposed to modify this piece of DNA at this time with this signal, and it gets changed so that now it's going to do something else. Well, what else is it going to do? Is it going to modify this guy over here or this over here? Or is it going to remove methyl groups? And there's no...
Because it's a full integrated system that needs to happen, right?
Exactly. Yeah.
Just like when we were talking about the flagellum and the secretory system, because it needs all the backdrop of the protein that will be injected, all the mechanisms, the idea that's going to use it for prey, etc. So it's not just one change that needs to happen.
Yeah, that's right. That's right. So Darwin's mechanism might be able to guide things one step. You know, what's the best thing right now, and whatever arrives first. But it has nothing to guide it after that if you've got to have two things that are coordinated. And even if you're starting with a sophisticated genetic engineering system, and certainly cells have sophisticated systems, if you don't have intelligent direction, that it's not going to build anything beyond what it's already got. So unless
we assume that it is part of the original engineering of the system, that there are sophisticated adaptive responses that then make the question even harder for the Darwinian mechanism, that the system is built so intricately to the extent it can have those responses with a complete picture in mind.
That's true. That's right. If there is even more intelligence built into the cell, well, okay then. But if it's on its own, if it's going to rely on a random change, then there is no evidence to see that it'll do anything beyond what we would think of as normal Darwinian processes would do.
Because if I understood what you meant, if there is 3 steps or 3 changes that need to happen, and if the first one happens by the Darwinian mechanism, obviously the other 2 will not happen, then natural selection will actually kick out the first one because it has no use. So the very mechanisms proposed by Darwinists themselves actually act against them to the extent that natural genetic engineering cannot be a thing.
That's right. That's correct. And it's also interesting, as I point out in Darwin Devolves, that if you need 2 coordinated mutations, you get the first one, and now you're kind of twiddling your thumbs until the second one comes along. That gives these degradatory mutations all the time in the world to occur. And
to
if
get rid of the first one.
Any of them helps a little bit, it will be fixed in the population in a very short time compared to how long it takes for coordinated mutations to arise.
I think this is your waiting time argument, right?
Yeah, that's one of them.
We have plenty of questions on waiting time, so we're gonna get back to this. And I think we are almost done with our primary questions. I hope you're not so tired, so you can answer some of the audience questions.
Yeah, I'll be happy. All
right. So let's go to the final ones. There has been this paper, an amazing one, and a very unamazing response from Darwinists to it. Well, actually, a very amazing one, an astonishing one. So it's about this botany research that was done on this one plant or category of,
Yeah.
where like it has been shown statistically that mutations are not random. What does that say? Can you give us shed some more light on that? And what does that say concerning what was before claimed to be unguided, that mutations are completely random and blah blah blah, etc. Well,
Okay.
what was the results
Well,
and what does it mean?
okay. Well, of course, the initial Darwinian thought is that mutations are random because mutations don't care about where they occur in a genome, and they just depend on physical processes. But there might be some regions of the genome that have a different mutation rate than others simply because they have a different physical characteristic. Maybe they have more AT base pairs, others more GC base pairs. But beyond that, there's nothing. This new paper in Nature looked at the distribution of mutations in the genome of this little weed called Arabidopsis, which is pretty common. And they looked at seeds of it, and they sequenced the DNA in the seeds, a bunch of different seeds, and said, okay, compared to the parent plant, what mutations have occurred just in this reproductive cycle, just this one reproductive cycle in the seedlings? And they showed that the mutations occurred more frequently in areas of the genome that don't code for proteins, or not genes. They're just connecting DNA, or they're other things. And they showed that in genes, in active genes, that there is maybe a third less, threefold less mutations than in the other regions. And what's more, they said if you look at genes which are indispensable, ones that if they were broken, the organism would die, they had even lower mutation rates. So it's even lower than general genes. So it seemed to be that these regions are protected from mutation. And it's kind of roiled the internet for a little while, especially folks who discuss this. And but I myself don't think it's a big result, because it's just not specific enough, and it doesn't change the mutation rate enough to actually affect much of anything. First of all, they propose that this actually developed by Darwinian processes.
That's the absurd part, actually. So random mutations are random mutations, even if they are not random mutations, because they are generated by random mutations. So come on.
Well, if all you have is a hammer, everything looks like a nail.
Yeah.
Yeah. But so the reasoning goes is that, well, if over time if there were mutants where repair machinery would be stationed closest to genes, then those mutations in those genes might get repaired very, very quickly, so that the genes would suffer less mutations than they otherwise would. So they gave it some sort of Darwinian spin. But the interesting thing to me is that it's only a threefold difference. Now, you should realize, or listeners should realize, that the mutation rate in multicellular organisms is roughly 1 in 100 million. That is, for every 100 million nucleotides, subunits of DNA that are copied, you make one mistake. And so if you were looking for a particular mutation that to arise to help in some selective conditions, you need 100 million organisms before, on average, you would get to one.
Wow.
Now they're saying you would need 300 million to get to that mutation. And but if you need it only there's no specificity other than in the genome as a whole. So it's not looking to, or it's not going to help on whether things can be positively changed or negatively changed. It just says we're going to keep this as it is. It's going to slow everything down within the gene area. And what's more, if you, within that, as I write in Darwin Evolves, in the gene, even after 300 million instead of 100 million, you're still a lot more likely to have a mutation that breaks the gene rather than improves it. So, and if you need two particular mutations, you've still got an astronomically bad chance of doing that. Again, I think it's one of these results that tell against Darwinian stories, but don't affect intelligent design very much. Or so I think it got a lot of attention, but I myself don't think the result's interesting, but I don't think it will affect these conversations in the long run.
I would like to propose something to you and get your opinion. So the question is, what does that suggest concerning the origin of complexity and life, or reducible complexity, in your opinion, as a creation model? And what I would like to suggest to you is that, and correct me if I am wrong, for example, if we look at the Venski experiment, and other places where we are just surprised that some mutations look very lucky to be random, like this transposon that made the citrate transport possible, that Venski called an exaptation. And as those things uncover, it just says that there is so much in the cell, more than what we know, including like a program that would say, this part, don't touch it, mutations go away, and this part, okay, you're allowed, but not so much. All right. And what if this is actually a program mechanism, so that there would be some little 1 in a trillion chance that this transposon will take the citrate transport into the right promoter, and it would happen given some environmental conditions will select to it. Mm-hmm. So it is, I don't know if we can call it epigenetic or something else, but wouldn't that actually, if more unfolds in this direction, show more of intelligent design? Because obviously the designer is putting controls to protect his design. The sensitive parts you want to completely protect, or further protect, threefold protect, and the parts where he would like to allow some little change so that some accidents can happen, then maybe in some time we'll find that they are not accidents at all when we explore more. What do you think about that?
Yeah, that's certainly possible. And of course, the more integrated and the more deeply design extends into the cell, we judge, as philosopher Thomas Reid said, he's a Scottish guy in the 1700s. He said, we judge the intelligence of a designer based on what he designs and based on what skill and stuff it takes. So the more and more and more skill that it takes, well, then our appreciation of the intelligence of the designer goes further and further up. That's certainly possible, and there might be all sorts of subtle things going on in the cell, including those. And again, I think it's a problem for Darwinism, because if you're Darwinian, then how do you explain how critical components of a cell developed in the first place if they're being protected by natural selection? Natural selection is preventing the mutations from
occurring there.
Yeah, exactly. And so even if you get a minimal function, which is critical, then if you you're going to have to protect it, and then you can't evolve it further in a Darwinian fashion.
Yeah, absolutely. So it again shoots itself in the foot that if those parts are protected, then how did they evolve in the first place? That would be another big problem for it.
Yes,
yeah,
exactly.
So, Dr. Behe, this was like a blast, and I hope you're not so tired, but thank you for accepting to take some questions from the audience. So let's go there. I asked them to tag me there, so we should find them easily. So here we go, guys. Those who are waiting for the questions, now it's time for the Q&A. So I'm going to the comment section. I hope you've tagged me. So yeah, wow, we have plenty of time. Okay, so we have fast enough. We have Dr. Faith says, is microevolution a fact where new information is generated? I think we actually answered that it is primarily evolution, so I think we can skip this one. And then, does antigenic shifting and drifting and super antigen are proofs of Darwinian evolution? I don't know what is antigenic. I think he's referring to genetic drift. Is genetic drift proof for the Darwinian evolution? And why neo-Darwinism is wrong? I think why neo-Darwinism is wrong was the topic of the whole conversation. So I think 1 and 3 are already done. How do you see genetic drift in the formula? Is genetic drift part of the Darwinian mechanism, or we can just call it part of nature, not necessarily part of Darwinism?
Well, it's a matter of definitions. I myself think drift is perfectly able to be fit into a Darwinian framework. In Darwin's theory, it has a mutation can occur, and it can be detrimental. So it can be cause harm and be selected against, or it can be favorable and it can be selected in favor. And some are more stronger than others, so they're selected or deselected more strongly. And in order to kind of quantitate that, evolutionary biologists use something called a selection coefficient. That is how strongly, and it goes from 0 to 1, where 1 is that it's selected entirely because an organism without that mutation dies. And 0 is essentially it's not selected at all because I'm sorry, we can go from +1 to -1. -1 would be it's a killer. Just the mutation occurring by itself kills the organism. And in the middle there is 0, and that's a mutation that doesn't have any effect. It doesn't help, it doesn't hurt. And so I think you can just consider these neutral mutations, drift, evolutionary drift, to be on the spectrum of possible mutations that Darwin proposed that has a selection coefficient of 0. So I don't think they need a new theory of their own, and I don't think they help at all. So so a lot of people spend a lot of time on this, and they jump up and down and say neutral theory is the big answer to everything, but I've never seen any evidence of that.
How can something that is neutral explain something that is progressive? Is a fundamental question, actually. But anyway, so here are some good, nice comments. So Salahuddin says, glad to see Dr. Behe. And Elias says, greetings, Dr. Behe, the legend. And I agree with him on that one. You have some good fans here. So, Ishmam says, what would Dr. Behe say to those who reject theistic takes on these issues? I'm not sure of what he means, but I think if there is obvious design, and then somebody like you and me come and propose that God is the designer, and then somebody goes to him and says, hey, this is unscientific. Is this a fair reason to reject that the designer is God? Is God unscientific in your opinion?
Well, yeah, I would say that intelligent design, as we emphasize, does not. The argument doesn't reach all the way to God. In order,
yeah, but if I propose that God is the designer, what's unscientific in that? If I propose that the sudden infusion of information is God infusing information, can
okay,
somebody else come and tell me this is unscientific to say God? Is it a taboo or something?
okay. Well, yeah, in a sense, it is unscientific because you want to have, if you define science as showing empirical evidence for something, and you said that God is the designer, okay, what evidence caused you to decide it was God instead of an angel or a space alien or some other possibility? You know, those they might seem far fetched to us, but other people might say, well, hey, maybe that's a candidate. There are certainly people who believe in aliens and in other worlds. So the question is, what evidence do you bring to it? I myself think that in order to conclude to a transcendent God, some beyond the universe, beyond the laws of nature, then you need philosophical and theological arguments as well. So science is just a branch of reason. It's not the totality of rationality. So I think we would need to use those other branches of knowledge than just science. But I think that science can conclude to design simply because we conclude design from empirical evidence. We look at the structures of molecular machines. We look at the structures of a mousetrap or Mount Rushmore or something. And we discern the purpose. That's a rational faculty that we have. We can see purpose. We have minds. We know that minds have purposes. But the question of who designed that is a separate one. If I asked somebody who designed Mount Rushmore, who had never heard of it before, they wouldn't know. Maybe God did it, maybe, but maybe somebody else did. So I think the difficulty is that if you're going to make a particular claim for a particular designer, then you have to have reasons for that. And if your reasons are philosophical and theological, that's great. Those are great reasons. But in order to qualify, I think, as scientific, if if Earthlings sent a spaceship out, which landed on another planet, and we met these advanced aliens, and they said, yes, we planted life on Earth, I could consider that scientific evidence. I don't think it's actually true, but it would be empirical evidence. I myself agree with you. I think God is the designer, as I've always said, but I think that's a different plane of reasoning than science.
So I think what your answer means is that if we're looking only at science as defined by being empirical, and this is actually a discretionary definition that people just agreed upon, it is completely different from looking at knowledge or epistemology as a larger umbrella, where knowledge can be taken not only from science, but there are other branches of knowledge that are outside the reach of science. So however, let me ask you this. If we're looking for the origin of life, I don't think that aliens can be an explanation unless we say that we're looking for the origin of life on Earth. Because if we are looking for the origin of life at large, then if aliens are living, then we have answered to nothing. And since the universe has a finite lifetime, it means that 13 billion years ago, there was no aliens because there was no universe of 13.8 billion. And then life must have started sometime after 13.8 billion years. It's even much shorter because the first stars have burst into oblivion. So we don't have that much time before the life of the Earth, maybe a few billion years more. And then the question would be, if it's aliens, then how come they are there? And if they are so advanced to the extent that they spawned us, it means they needed so much more time to evolve than us, set aside reaching to our planet. with all the problems with cosmological distances.
Sure.
And then we will have to resolve that it cannot be a material living being, because then it has an explanation. And then philosophically, we can argue that this being has to be beyond this universe. And if we define God as that, then the answer is God. I don't think science can stand to this kind of argument. It just needs a little bit of logic on top of it. I think.
Sure.
Is that what you mean when you?
Yeah,
when you
I agree with it. I agree
Yeah.
with everything you say, but I'm, thinking of the philosophy professors, sitting in their office and saying, well, maybe in a different universe, there's a physicist which, makes black holes, and the black holes expand, and there's lots of crazy things out there, especially when you read academic writings. So in order to avoid arguments like that, I usually say that, well, we can clearly conclude life was designed, and, we can everybody can decide, who they think is the most likely candidate for the role of designer. And most people will come down, I think, to the same conclusion as we do.
Yeah. So we have another welcoming. It's from CIID. I think it's an Italian friend. He says, it's always a great pleasure seeing and hearing Professor Michael Behe and his scientific truths. And then he says, a heartfelt farewell from the Italian Center for Intelligent Design. So yeah, it is an Italian friend. Okay. So Arshad says, looking forward for a good discussion. That was truly at the beginning. Okay. Ilyas says, my first question is, I heard years ago that Lenski experiment is shutting down soon. Is that true? Do you know about that?
I've heard him talk that he wants to give the experiment to another scientist to carry on, but he's getting older too, like the rest of us, and so he wants to pass it on. I don't think he wants to shut it down.
All right. Salahuddin asks, finding truths, if intelligent design works, how harmful mutations happen? I think we've explained that when we talked about devolution already. And then Ilyas already says, it's nice that we have already answered plenty of the questions. How would an experiment involving microbes with enormous population sizes and fast replication apply to organisms like humans or whales? Wouldn't population genetics preclude this? Do you have a comment on that?
Yeah, that's an excellent point. That larger organisms like humans and whales have a lot smaller population sizes, a lot fewer of them. That means there's fewer opportunities for mutations to arise, and their generation times are decades, not 20 minutes like bacteria. So even the things that are possible with bacteria are ruled out for larger animals. And even bacteria don't really do very much at all, then we can conclude that pretty much all the features of larger organisms were intended, at least except things like the shape of your nose, perhaps, or something something minor.
Yeah. So if bacteria with the tens of thousands of generations in the Lenski experiment cannot do that much, then mammals that have ages in the tens of years can definitely almost do zilch. Yeah. So we have Nafisa Pandalangiri says, can you explain RNA hypothesis? What are the merits, demerits of the hypothesis? I think this is a big discussion.
Yeah.
I would point you to a discussion that I had with Dr. James Tour, where we discussed origin of life. But if you want to give a little comment on that, Dr. Behe, just go ahead if you'd like.
Okay. Well, yeah, sure. A big problem for science is, how did life get started? And the short answer is nobody has an idea. But people propose that RNA, which is similar to DNA but has a slight chemical difference, people propose that somehow arose first in an inorganic world, because RNA can transmit genetic information like DNA, at least theoretically. And also, RNA can fold up on itself and form structures that have chemical catalytic activity, like proteins do. And proteins are actually the machinery that gets the work of the cell done. So, in some folks' minds, if you have this, then you can cover both bases. You can have things that machinery that works, and you have a genetic carrier. Problem is that RNA is extremely fragile, that nobody has really gotten it, has a clue how to synthesize it under plausible prebiotic conditions. And even that, there's still no answer to the information that the RNA would require. There's just like with arrangements of letters, you can arrange the nucleotides of RNA in a bunch of different ways, and usually they make absolutely no sense, and it's only rare instances where they would make sense. And so how do you get those rare instances and coordinate them with other RNAs that would have to do other things? So it's in short, it's just speculation, which has very, very little evidence for it. The only thing that one of the reasons it was suggested is that in modern organisms, there is RNA that does some
mediation between the DNA and the proteins.
That's right.
And autocatalytic functions.
Exactly. Yeah. So it has catalytic activity and can carry information. So people kind of projected this back into the beginning of life and said, well, why not RNA?
But in the context of irreducible complexity, I would because they would then claim that RNA spawned the DNA replication, transcription, translation, central dogma. But to me, correct me if I'm wrong, and I'm doing all this series with Dr. Fuz, and we're really dwelling on DNA. What we've seen over and over again is that the DNA genetic system is irreducibly complex. If you just remove one aspect, it just doesn't work. And then the question would be, how would RNA transform into DNA if DNA is irreducibly complex? Because then we'll run into exactly the same problems that you have explained. Set aside the devolution properties of RNA. Would you agree to that?
Sure, absolutely. Yeah, there are many, many different things are required to get life started. So it's irreducibly complex. If you need all of these different things, and then without even one of them, it wouldn't work. The nice thing about origin of life is that you get a lot of scientists agreeing that science has absolutely no explanation for it. Unlike for evolution, which they still don't have an explanation for either, but they think that they do. So yeah, that's good. It's interesting. There's a man named Eugene Koonin who is a very well-regarded scientist, and he wrote an essay where he says that since the origin of life is so difficult to imagine, probably we live in an infinite universe, and it just by accident, it all came together here on Earth. So that's as far as I'm concerned, that's the abandonment of reason. He's just saying, well, it just popped into existence by chance, not by intelligence, just utterly by chance. So that's
Darwinizing the genesis of the whole universe in this case, or of our universe, actually.
Yeah, that Darwin was one smart guy, I'll tell you.
Yeah, not too bad. There is this interesting question about cancer. So it says, what does it mean by degradation of genes? For example, in case of proto-oncogenes or tumor suppressor genes, it can mean very different things. I think he's indirectly referring to the fact that cancer does all those tricks. Some of them look innovative, and especially in countering, chemotherapy, etc. What's your take on this?
Well, first of all, in view of intelligent design, does not say that nothing can go wrong in design systems. an automobile is designed, that doesn't mean you can't crash the automobile or that it can't get a flat tire.
Yeah.
So we know that mutations happen. We know that, and the environment can affect us. We live close to New Jersey here, and they have these big chemical refineries, there's more people get cancer there than elsewhere. So genetic accidents can happen, and that's essentially what cancer is. So some things break. Your cells have to be kept under tight control so that they integrate with the other cells in your body. And if you break the control mechanisms, then they can start to reproduce when they're not supposed to reproduce and cause tumors. And what's more, when chemotherapy is used or other approaches, it's oftentimes the case that they can escape those poisons or those treatments by breaking another gene, just like the polar bear did. If a chemical, if a therapeutic chemical affects a particular gene and it can live without it and gets rid of it, then the tumor can continue to grow even with our best treatment. I should say that I think Darwinian processes or random mutation and selection is a great explanation for cancer. But that just goes to show you what random processes do when
they do cancer.
Yeah.
Too bad.
When they're given their way, they reduce information, they mess things up, and I would never listen to somebody who touted that as a plus for Darwin's theory of evolution.
Wow, that's really a great insight. We have Ilyas here talking about something called the Million Mutations Project, and he says it's like Lenski experiment but with worms, and not a single beneficial mutation was observed in the colony. Do you have a comment about that? Did you hear about that experiment, Million Mutations?
No, I haven't heard of that, but I'll write it down and look it up as soon as I get the chance. The Million Mutation Project.
Yeah. So this is a question about mutations are not entirely random. Actually, we addressed that in the botany question. Have there been any developments on waiting time problem, aside from making it worse via slower chimp mutation rates since Duret and Schmidt, or do they still concede it? Laughing out loud. So is there any further work that that you're doing on the waiting time problem? You explained it in a nutshell when we were talking, and you explained that you will need that is so small, the mutation rate is so small, so it's 1 in 100 million, then you need so many organisms, and then if one good thing happens, then by the time the next thing happens, the first one would have been already destroyed by either a further mutation or by natural selection. So are there people who are really looking into waiting time and doing research on it to further develop the argument?
Well, I am not right now. I talked about it in Darwin Devolves somewhat, but there are folks, colleagues of mine who are working on it, colleagues from the Discovery Institute, and have published a couple of papers in the past several years on it. A woman named Anne. I was getting old here. A man
not
named
at all.
Oya Hasier and Ann Gager and a number of other folks too. As a matter of fact, Gunter Beckley is a co-author of the paper too, who's been a guest on the show. So if you do a search on any of their names, you'll come up with these publications, and these are scholarly publications, so they're heavy on math and stuff. So be prepared once you get the paper.
All right. We have a comment here from SpongeBob Imagination. Says, great discussion. Okay. And he says he has a question for you. He says, someone born and raised in a polluted area won't notice the stench like a tourist would. Is something similar happening to those raised uncritically on evolution? I think he's referring to the almost what we can call indoctrination in the biochemical and biological fields of study, which is really miserable. What's your advice to people who are going to go into these fields to study them?
Well, it'd actually be great to go into them because we need folks who can think more broadly, but just make sure to read with an open mind criticisms and other views of evolution, the development of life. And I think you'll find, as I did, that the more you know about biology, the more and more you see the design, and the more and more difficult it gets to think that a Darwinian view is correct. So
isn't that your story, Mike, yourself?
Yes,
that actually marveling on what you have learned is what actually brought you to faith.
That's correct. Yeah. I used to think Darwinian evolution would be true, not because I knew how it could explain anything, but because my teachers told me that it was true, and all the smart people I knew thought it was true, and so therefore it was true. And so, and somebody going into it, like the last questioner, you have to be ready to resist the popular opinions, the social pressure. But we can certainly use you if you're willing and able to join us.
Yeah. So here we have somebody commending your health. He says you look good. And then, yeah, he's asking you a question. So we wish you really long life and great health. And he then asks, could you summarize your mathematical argument against Darwinism from the Edge of Evolution? And he says it's an amazing book. By the way, everybody or everyone should read it.
Mm-hmm.
So I think he's talking about the waiting time, or is there something else there?
Yeah, that's a waiting time argument. Essentially, it says how long, if you need 2 mutations to have some selective effect, how long will it take that to occur? And it turns out that if you only need one mutation, Darwin's mechanism works just great. But if you need 2, then you're already in big trouble. And I go through some arguments, especially ones concerning malaria, where some drugs, in order to acquire resistance, the malarial parasite had to have 2 particular mutations, and it took them an enormous number of organisms, cells, to develop this. And I write that this would be beyond the ability of any mammalian creature or larger vertebrates. So, the Edge of Evolution just sought to say, what's it reasonable to think Darwin's mechanism can do, and what would require purposeful design? And the conclusion is that Darwin's mechanism doesn't explain too much, and that design went deeper into life than I had thought, even in Darwin's Black Box. And I should say, in the newer book, Darwin Devolves, we can conclude that it extends even deeper, and that Darwin's mechanism explains even less. So you can see where the trend is heading.
Yeah. I think this question has already been answered from Ali. He says B uses the bacterial flagellum for irreducible complexity. Type 3 secretory system is supposedly a newer part of the gene with less parts. Isn't that so? We actually explained that. And actually, he listened to our explanation and says, interesting, thank you. Yeah. And the part that actually the type 3 secretory system appeared after the flagellum was really a blast. Then Professor Another Letter says, question for Dr. Behe, if hypothetically a protein was randomly generated in a primordial Earth, what is the most likely outcome for it? I'm not sure how it will be generated in the first place, but if a protein just happens to occur, what's its fate?
Well, the most likely outcome is that it would be degraded and go back into the oblivion at the bottom of the sea.
Great, Dr. Behe. This was really great. I think number one, thank you very much for coming. And it's really an honor, and it was really so much fun talking to you, and so informative, and thank you for being the person you are. I would like to close by telling you this. In the Bible and in the Quran. So I think in the Bible there is something in the lines of God commanded the earth and the seas and then animals and plants came to existence. It's something in those lines. So there is this clear assertion of an intervention and a purposeful and intent and design, and from the first point there is the foresight of animals and plants. And there is something, there are many ayat or verses in the Quran that they say the same thing. And there is also this ayah in the Quran that says, the translation of meaning would be, we will show them our signs in the horizons and within themselves until it is evident to them that it is the truth. And my question to you is, how come some people would know what you know and what other biochemists know, and they see the intricacy and the greatness and the miracles in ourselves? And I would expect that in ourselves would ultimately refer to molecular biology, to the real inner workings of our bodies, where we are the pinnacle of creation, obviously. How could somebody see this and not come to God, just like you came to God, you came to believe? And actually, I was having this discussion with Dr. Faz Rana on Thursday, and he had a similar story, and with Dr. James Tour and with others. Maybe Dr. James Tour has other reasons also, but so many that I have talked to. How come there are people who don't really reach this conclusion, in your opinion? And we close with this, if we can take your take on it in a couple of minutes.
Well, again, the short answer is I don't know.
From your discussion with them, when you sit with your colleagues, guys, how did this come into being? And they say, we don't know. So why don't you believe in the ultimate designer? Why don't you believe in God?
A number of them say, well, I'm a scientist. I search for rational explanations, and they have this pride of being smart and so on. Other folks just don't want there to be a God. They just don't want. There's a famous philosopher named Thomas. What's his name? Thomas. Anyway,
Thomas Aquinas. You mean Aquinas?
No. A modern philosopher from Yale University.
Okay.
Yeah, his name escapes me again, but he wrote a book called The Last Word in 1990, and he said that he himself, he doesn't want there to be a God. He's an atheist because he doesn't want the world to be like that. And
wow.
but interestingly,
at least he's honest about it.
Yeah, and he's actually a real skeptic of Darwinism, and he's hosted intelligent design events, but he does not. He's looking for some natural explanation. But he's trying to, have the universe have a mind or something, a pangenesis or something. But, some people, for reasons that escape me, just have such an attitude. And there are many scientists who just look with contempt on religion and have that same attitude. They don't want there to be a God, but it's because, they want to decide for themselves how to live their lives and figure out all the mysteries of life and the universe for themselves. So it's a sad situation, and I think you'll agree that it's not hard to see these design in this intricate machinery of the cell, but some people don't. So,
wow! Even if their fate that for eternity depends on it, they just don't want to look. They just look the other side. That's it.
Yeah. Well, Unfortunately, yes, that's the case. People can be very, very stubborn.
Yeah. So, Professor Dr. Mike Behe, this has been so great. Thanks 3 billion for your time. 3 billion being the number of base pairs in the human genome. So one thanks for each base pair. This has been really great. May God reward you for your great contributions to both science and faith. And thanks for everybody who's been here to watch this live. I have a request from you. Just share this to the maximum number of people that you can share it to. If there are people who have put some extra questions, I try to stop it at 3 hours because with Dr. Behe has been so generous, and definitely we can't thank him enough for this. For everybody who's been there, thank you very much for being here. And for those who are not, please share this and do your share in spreading the truth. I would now put the extra and just ask you, Dr. Behe, to remain for one more minute to give you a special thanks again after this. And for everybody, have a blessed day and great time with your friends discussing what we have discussed today. Assalamu alaikum wa rahmatullah. Peace be upon you all.